The hydroxyl group of an alcohol can get in the way during a synthesis. Protectant groups offer a solution to that problem. Here's a fun example. Take a look at this. I've got a molecule up here that is a fatty acid. It's a type of molecule that occurs naturally. Plays both physiological roles in plants and animals and roles in terms of our nutrition. It's entirely plausible that we'd like to synthesize this particular fatty acid, which you see has a carboxylic acid at the end and a cis double bond, also called Z, along the chain. We need to do three things. We need to build up the long chain, we need to put in a cis double bond, and we need to make a carboxylic acid at the end. Knowing the chemistry you already know, we're in a good position to make that happen. This alkene with cis stereochemistry can be made from an alkyne. You may recall that requires the Lindlar catalyst with hydrogen gas. Very good stereoselectivity. We've just learned that carboxylic acids can be made by oxidizing alcohols. So let's think about an alcohol as a precursor for this alkyne. And we know that long chains can be built up by alkylating smaller alkynes. So that brings us to the thinking that maybe we should use two pieces that look like this to make the longer chain. We picture this pair of electrons of the alkyne being used to form a sigma bond with that carbon as it displaces the bromine. This is an SN2 reaction. But wait, we're proposing to use an acetylide, a strong base, in the presence of an alcohol, which has an acidic hydrogen. So we have a very strong base, we have an acidic hydrogen. Those two things are not going to be compatible. They'll react with each other. Those electrons will remove a hydrogen from the alcohol. And our reaction plan is dead in the water. So, as I mentioned at the beginning, sometimes hydroxyl groups get in the way of synthesis plans. There's a clever way to solve this problem. Because these two species are not compatible with each other, let's just not plan to use this acetylide anion in the presence of an alcohol. So let's use an alkyl bromide that has what we call a protecting group attached to the oxygen in place of the hydrogen of an OH group. Now there's no acidic hydrogen. So now we could plan to do our reaction where we have the acetylide being alkylated by the bromine to make a new carbon-carbon bond and build up the long chain. What remains to be done is to convert this compound to the alcohol. So we've used an alcohol, converted it to something else that provides a protecting group. We've done the chemistry that can't be done in the presence of an acidic hydroxyl group with the protecting group there. And then at the end, we've removed the protecting group. What remains, of course, is to figure out what that protecting group could be that works well. Well, in fact, there are a few choices. Here's a good one. Starting with this bromoalcohol that we wish we could use but can't, we'll put what's called a trimethylsilyl ether attached to that. TMS, trimethylsilyl, is this guy here. 3 methyl groups attached to silicon, and the silicon is attached directly to the oxygen. These TMS ethers can be prepared in very high yield and have no acidic hydrogen. Then we do our chemistry, reaction with the acetylide anion to make the new carbon-carbon bond with the TMS ether attached. And following that, we'll remove that TMS ether with a special reagent that cleaves the silicon-oxygen bond. A reagent often abbreviated as TBAF, and you'll see why. TBAF is tetrabutyl ammonium fluoride. It has 4 butyl groups attached to the nitrogen, which makes this an ammonium group and a fluoride anion. And it's the fluoride anion that actually does the chemistry, cleaving the silicon-oxygen bond. When you finish with adding water, you make the product you want. So we have a clever solution. We'll use a protecting group to remove the alcohol from the presence of the alkyl bromide. Let that alkyl bromide, as a TMS ether, react with the acetylide anion, make the carbon-carbon bond, and then remove the protecting group. Protecting groups are common in organic synthesis. It's a clever way to remove groups that otherwise would be involved in a reaction when you don't want them to participate in it.